Hello, I'm Felipe. I will present this project entitled Patterns and Determinants of an Atypical Dengue 2 strain isolated in Brazil. I'm supervisor is Dr. Camila Malta Romano. This project is a side project, not my master's degree project. In Brazil, dengue is epidemic, and each year we suffering with dengue, and now 4 serotypes co-circulated in Brazil each year. Esqueci de tirar o mapa. Eu vou me matar. In our laboratory, we isolated dengue virus strain, JH, of a case of dengue without warming scenes. Based on a small NSC1 fragment, this virus was classified dengue virus 2. Interestingly, the virus is neurovirulent in mice with 0.1 MLE, leading to death 3 days. And we studying this strain dengue virus, and we compared it to other strains obtained for a dengue without warming scenes, and this neurovirulence and the hematological patterns of mice are different. And for example, this innate JH will kill mice at 3 days, and here, and in B, clearly see that the replication is faster. In C, JH is drastical, increasing the lactate dehydrogenase, and in D, we see that JH infected mice lost weight. The hematological patterns in mice is have disturbed in JH infected compared with American Asian isolated from a classic case. We then decided to study this strain in more details. The disease was subjected to next generation sequence using the ion torrent from 3 amplicons completely sequenced the viral genome. This data we analyzed to infer the viral genotype, through the phylogeny, and map the mutation sites related to neurovirulence. Using maximum likelihood, the JH strain was determined American dengue 2, but was very different from another virus from the same genotype. This is an American genotype, and JH was here. To determine in phylogenetic analysis, we used 208 reference sequence of all dengue 2 genotypes, and JH close to 353. To determine the precise level of divergence between JH and all dengue 2 virus, we built this graph in red here. We can see an average distance among American virus with about 3%. And in blue, we can clearly see that JH divergent from another American by 7%. And this is between the divergent observed for American, Asian, and cosmopolitan genotypes in relation to America, which is around 10%. Only for comparison, Sylvatic strain diverge 20% from American genotypes. After characterized phylogenetically and size, we already know the full genome. Experiments were conducted in order reverse neurovirulence phenotype. The neurovirulence JH was consecutively passed to mosquito cells, C636, until it was not longer neurovirulent. We tested the ability to kill mice on every 8 passages. In the passage 23, the neurovirulence decreased, but not affecting 100% of mice. In the passage 29, the JH virus completely lost its neurovirulence. This graph represents the decrease of mortality in the indeterminate passage 23 and in 23, and this passage 29 is survivors 100%. The non-neurovirulent strain was then sequenced by the same method applied to neurovirulent strain. The genomes were compared at older to identify change in sites that would suggest the neurovirulence characteristic. These sites were subsequently compared to those already reported as responsible for neurovirulence in other dengue virus. This table shows all the change observed at time 0 and time 29, and their frequency are deep sequence. A total of 15 changes were observed and 8 non-synonymous. The last column shows the consensus of the respective position for the non-virulent virus of the same genotype. The most important mutations are these, there, and here. Phenylalanine and leucine in position 402 in the envelope. That's mutation. It's that mutation appear in NGC dengue virus. The original JH have a deletion at the position 539 in of the NS3 and arginine, which was recovered in the passage 29. And this region is present in all American consensus. And two other non-synonymal changes in the NEC5 genes were observed. This is ongoing project, so the perspective are to finish sequence to passage 23, and to really determine the sites responsible for the virulence. We are adapting the virus from passage 29 in mice again. And the questions are no. Do we expect that the virus in fact to become neurovirulent? If so, do we expect it to see changes at the same sites? And how much the phylogenetic characteristic we observed influence at the phenotype? Well, good luck. I'm nervous. And special thanks of Doutora Camila Malta Romano group and Dr. Luis Carlos group of IMT Virology Laboratory and ICB Virology Laboratory. It's all.